So I'd like to continue from my defense and talk about developing DNA-based diagnostics for visceral leishmaniasis and other neglected tropical diseases. So yeah, if you want to develop a new point of care test, there are basically three biomolecular methods that you could develop. And the first is immunological POC tests. So that is very similar to the RK39 test that Dr. Rittmeyer has been speaking about. Basically, you get an infection and your body produces antibodies against the infection, and that's what can be detected on the test. So the advantages is that it's a very developed format. It's widely available and widely accepted. However, it is prone to false negative test results. As you've seen in the first talk, people's immune systems react differently, but also that antibodies persist post-treatment. So another option would be a biomarker-based POC test, and I would definitely say that we are in the biomarker discovery stage. Fortunately, I think one of the advantages is that synthetic aptamers are very cost-effective in order to detect a biomarker once it's been identified, but there are definitely higher initial costs for development when you want to make this commercially available. And also a lot of further development is required for field deployability. So it's always great to have a proof of principle, but then all of these need to be tested and validated in the field where they'll be used. And another option is nucleic acid based, so that's DNA based. And it builds up on the principle of PCR, where you can amplify the DNA of the pathogen itself. So the advantage here is that you do have test of cure capabilities, so it's confirmatory. However, the same with biomarkers, there's a higher initial cost. So far, it's been restricted to a laboratory, and so you need further development. But given the expertise of the group and my background as well, I chose to focus on developing a DNA-based point of care test. So we use CRISPR-Cas as our DNA detection tool. And I'm sure you've heard a lot about it in the news, especially after the Nobel Prize was won this year for chemistry. So CRISPR-Cas, it is a bacterial adaptive immune system. So much like humans have an immune system that protects us against viruses, such as COVID-19, bacteria also have an adaptive immune system. So it's quite a complicated system. This picture, I will just like to point out that although there are many components, we can repurpose the components that we need for just DNA detection. And one such system is the CRISPR-Cas9 system. So how do we build a CRISPR-Cas9 tool for detection? Well, we start by looking at its individual components, and we start with the guide RNA. So fortunately, CRISPR-Cas is a very simple system. There are only two components. This guide RNA essentially is what will bind to your target DNA. So the red part is the part that binds to the target, and that you can change. So we can encode a sequence for visceral leishmaniasis, but we can also encode a sequence for another disease. So it's simple, but it's also very versatile. So this is one part. The black part is what's important for the second part of the system, which is to recruit the Cas9 protein. So together, individually, these two components don't do anything, but when they are in complex, they form a search complex. So two-component system, and basically it functions as a detection tool. So if you think of a sequence, a DNA sequence of a parasite, if you think of that as a barcode, you can think of the Cas9 search complex as a scanner that scans the barcode and looks for a match. But now, if the Cas9 protein happens to find a match in a sample, we need to link this to a colorimetric readout so that we can get a visual readout of the test result. And for that, we have slightly modified this Cas9 protein. We've added a trigger molecule, and the trigger is what allows us to see the results with the naked eye. So this is the system. It's only two components. It's very simple and it's very versatile. And now I'd like to take you through our molecular toolbox, and this is how we've developed our novel DNA detection scheme. So as Dr. Rittmeyer mentioned, it's very challenging to get a sample, especially in resource-limited settings. So if you have to do a needle aspiration, there are challenges, and it can also just be dangerous for the patient. So we aimed for a non-invasive sample procedure, and so something from a pinprick of blood or from a urine sample. And we've developed a one-step extraction. So basically we have this disc, it's a membrane, it's a matrix of different pore sizes. So it traps things based on size. And we functionalize it with a molecule called chitosan. And what this chitosan does is that when you add the sample to this disc, it entraps everything, the DNA, which is represented by these straight lines, but it also traps proteins and other cellular components. And in one simple step, by adding an alkaline buffer, we can release the DNA while separating it from the proteins and the rest of the components. But naturally, of course, this would release all of the DNA. So the genomic DNA from the patient's parasitic DNA or other parasites. So then what we need to do is that we need to specifically amplify only the target sequence that we are looking for. And for that, we use an isothermal amplification called RPA. And this allows us to, like, if you can see, there's only one green molecule and a few gray ones. And this allows us to amplify the green molecules only and to separate them from the rest of the molecules. And we do that with a biotin, streptavidin interaction. So now that we are able to immobilize only the DNA that we want, we can also wash away all the rest of the DNA that we don't want. And now that we've amplified, so we've made many copies of the target sequence, we can use the CRISPR-Cas9 system to bind to the target because that's what the native system does. And then for readout, we've coupled the circular template. So you can see the circle here. And this allows us to do a second amplification and to also give us the colorimetric readout. So for the rest of the presentation, I'd like to focus on steps 2 to 4 and take you through our validation of every step. So looking at step 2, when choosing a target sequence, so this is the specific DNA sequence of the pathogen that we want to detect, we chose to target kinetoplast DNA because there are thousands of copies per parasite. So this is an amplification within itself. We get to detect something where there are many copies, even in a single parasite. And what we did was to see if the sequence that we had chosen in silico was present in patient samples, in blood and urine samples. So this is a gel electrophoresis, and these bands represent DNA. So you can see that the sequence, 83 base pairs, is present in the patient blood and urine, but not in the healthy blood or the healthy urine. But you will also see more than one band. So you'll see that there are non-specific amplicons, but what's really great is that when you do RPA, it works, and there's a broad temperature range. You can just leave it on the bench top. You don't need any external equipment, and it works in as quick as 15 minutes. This part of the assay is suitable for any downstream detection method. So we use CRISPR-Cas9, but you could also adapt parts of this detection scheme for other downstream applications. But now that we know that the sequence is there in a sample, we wanted to couple it to the Cas9 system, our detection system. And so we do a couple of things. You see here that we add the RNA and the Cas9 protein. We make a search complex, as I described, and then we incubate that with the target DNA. This is another gel image where you can see multiple bands. In lane 1, you see that the band runs quite low. It runs quite low in the picture, and that's because the DNA is small. But when you add the protein to it, it makes the complex bigger, and this causes it to run higher up in the gel. So in lanes 2 and 3, you can see that the proteins have bound to the DNA. So we know that our modified Cas9 protein is able to bind to the Leishmania target. So the third step, which deals with sensitivity, is to now amplify the readout and to couple it to a visual colorimetric readout. So we do rolling circle amplification. This is just a qPCR readout. You can see that it works from 15 to 65 degrees. It has an optimal temperature at 40 degrees. But it's very nice that it has a broad range. It works in a broad range of temperatures, because if you do it here in the Netherlands where it's 20 degrees, or if you do it in Africa where it's 40 degrees, it will still work. And to couple this to a readout that's visible to the naked eye, we've used these G quadruplex structures. But yeah, both of these reactions, they work 25 to 40 degrees. You just need to leave it on the bench, and the color develops within 5 minutes. So we get sensitivity in our assay when we amplify the target with RPA and when we amplify the readout with rolling circle amplification. So these are the different steps that we check individually. And the next thing that we need to do is to see if they work consecutively. So this only now includes steps 2 to 4, so not the sample extraction. But basically, we do the RPA, we mobilize it in a tube, we add the Cas9 protein, we do rolling circle amplification, and we see the color readout. And we see a very clear colorimetric readout. There's a clear difference between a sample with the target DNA and without. You'll also see here that this hasn't been quantified, so it's just a qualitative answer whether the target is there or not. So we have a very broadly applicable biosensing tool that can be used for different applications. The specificity is there from the amplification and from the CRISPR-Cas9 detection. So when packaged into a device, we think it could be used for screening and for test of cure because it's based on detection of the DNA itself and not on the person's immune system. It's an isothermal reaction. It has a broad temperature range of operation and the readout is visible to the naked eye. However, it is currently an overnight incubation. We have only validated part of our assay with 1 patient sample, so there's a lot of work to be done. It's currently not quantitative, but I do think there's a lot of room for us to make it quantitative with further development. We are still looking at the surface binding capacity so that we can determine the limit of detection. And I think packaging is also very important for this to be applied in resource limited settings. And so we have something like this in mind. This is a microfluidic device. It's a closed system so that you don't have any cross-contamination issues, and also that it's just more automated. So keeping this in mind, you've seen the context from Dr. Rittmeyer's presentation as well, but these are the resource limited settings that we would aim for. This is where we would like our diagnostic test to work. But unfortunately, at this present moment, we still think that it needs to happen in a hospital or a treatment center where there is capacity to do pipetting and to do some of the reactions, but this is something that we are optimizing. And with that, I would like to thank everybody that's been involved, my supervisor, colleagues, collaborators. We've had lots of students, but also our stakeholders and especially our local stakeholders as well. So, are there any questions?